Okay, for the introduction, CRISPR-Cas9, known as clustered regularly interspaced palindromic repeats, is a gene editing technology causing a major upheaval in biomedical research. It makes it possible to correct errors in the genome and turn on or off genes in cells and organisms quickly, cheaply, and with relative ease. It has a number of laboratory applications, including rapid generation of cellular and animal models, functional genomic screens, and live imaging of the cellular genome. It has already been demonstrated that it can be used to repair defective DNA in mice, curing them of genetic disorders. And it has been reported that human embryos can be similarly modified. Okay, on this video, show that how CRISPR-Cas9 work into gene editing. For CRISPR-Cas9 timeline, for December 1987, the CRISPR was initially revealed. For January 2000, short regularly spaced repeats are additional clustered DNA repeats found in various bacteria and archaea. For March 2002, the term CRISPR-Cas9 was initially published. Okay, for November 2005, new Cas gene families are discovered by American researchers, and these genes seem to aid in defending against invasive bacteria. For March 2007, for the first time, experiments show how CRISPR and Cas9 genes work together to defend bacteria and viruses. And for the December 2008, the RNA gene silencing pathway connected to the CRISPR-Cas mechanism was published by scientists. For March 2011, Jennifer Doudna and Emmanuel Carpentier collaborated to study the Cas enzyme. December 2012, US Patent Office received expedited application for CRISPR-Cas9 technology. For August 2013, the rat genome was engineered using CRISPR-Cas. Plant genomes, including those of rice, wheat, tobacco, Arabidopsis, and sorghum have been modified using CRISPR-Cas. Okay, for August 2012, publication of a completely novel CRISPR-Cas9 system based on gene editing technique. For September 2012, researchers at Vilnius University have published a study clarifying how CRISPR-Cas9 can modify DNA. For January 2013, editing the human genome with CRISPR-Cas and zebrafish genome editing involved the use of CRISPR-Cas. Okay. For March 2013, Saccharomyces cerevisiae is a yeast species used in winemaking, baking, and brewing. Its genome is edited using CRISPR-Cas. For August 2013, enhancement to the CRISPR-Cas system's specificity. For December 2015, in a mouse model of Duchenne muscular dystrophy, the gene editing technique CRISPR has been effectively employed to enhance muscular function. June 2016, the first clinical experiment employing the gene editing technology CRISPR-Cas9 to treat patients has been approved by the NIH. October 2017, enhancements to the CRISPR technology for base editing have been revealed, enabling the modified of specific DNA chemical letters without the need to break DNA. December 2018, the effectiveness of first-line chemotherapies for lung cancer was partially restored by CRISPR-Cas9. In December 2019, Chinese scientists found guilty of altering human embryos using CRISPR-Cas9. June 2020, published research raises concerns about the safety of modifying human embryos with CRISPR-Cas9. October 2022, Emmanuel Carpentier and Jennifer Doudna were given the Nobel Prize in Chemistry for the development of a method for genome editing. For September 2022, Vertex has received approval from the FDA to submit a ruling application for assessment of a CRISPR-based treatment to treat beta thalassemia and sickle cell disease. For November 2022, CRISPR appeared to be a potential method for modifying immune cells to increase their ability to kill cancer cells, according to a small clinical trial. Gene editing tools for CRISPRs discovered in thousands of stages. November 2023, conditional permission for the world's first CRISPR-Cas9 gene editing therapy for 2 blood diseases has been granted in the UK.
Will elucidate the reason for the revolutionary of CRISPR-Cas9 breakthrough which has profoundly transformed the planet. CRISPR-Cas9 hold a great promise in treating genetic illness resulting from the specific mutation in individual genes. Some disorders that fall under the category are cystic fibrosis, Duchenne's muscle dystrophy, DMD, and hemoglobinopathy effectively rectify the prevailing of the Fc-causing mutation in intestinal organoids. They show that after correcting the mutation, FCTR function, which is responsible for conducting iron across the cell membrane, was reinstated. Also, treatment for HIV. CRISPR-Cas9 has potential to be used as treatment for infection disease, including HIV in a clinical setting. Antiretroviral therapy is a highly successful treatment for HIV, but it cannot cure the disease since the virus becomes permanently integrated into the genetic material of the host. The applicability of the CRISPR-Cas9 system in specifically targeting the activity of the HIV-1 genome. The inactivation of HIV gene expressing and replication was shown in many cells that can be latently infected with HIV without any detrimental effect. In addition, it is possible to immunize cells against HIV-1 infection. It is also engineering somatic cells ex vivo to treat malignant or other diseases. Is in increasing curiosity in utilizing CRISPR-Cas9 to alter T cell and stem progenitor cells generated from patients with the aim of the subsequently reintroducing them into the patient of therapeutic purpose. The manipulation of the T cell genome have previously demonstrated efficacy in the treatment of blood cancer and promise for the treatment of solid tumor, immunological defense, and autoimmune disease. Agriculture application of CRISPR-Cas9 genome in editing in the plant species. The genetic alteration of the plant, namely crops, can be used to modify and fine-tune specific phenotype or feature. The objective is to integrate and improve crops that can provide many benefits to the general population. The accuracy of CRISPR-Cas9 technology guarantees the highly dependable approach in the genome editing that can avoid generating unexpected modification in other parts of the genome. Furthermore, it also improves on quality of the crops. An important application of CRISPR-Cas9 technology in the term of sustainability is capacity to improve the quality of agriculture goods. The CRISPR-Cas9 technique has shown the many potential to improve specific mutation in the rice, which is significant food supply of the world population. Subsequent to this discovery, numerous endeavors have been directed toward comprehending the role of specific genes in examining the impact of the gene modification in the rice, with the aim of implementing the finding with practice manner. Limitation, several obstacles persist until the full potential of CRISPR-Cas9 may be realized. The development for viable medicine for the patient. An important concern resolves around the method of delivering gene editing to the specific cell, in particularly when the treatment is administered within a living organism. In order to transport Cas9 nucleus expressing gene and guide RNA into the living organism without any harm, an appropriate carrier is required. An additional notable problem arises from the potential occurrence of off-target effect on genomic region distinct from the specific region being targeted. Accidental modification of the genome may result significant and enduring issue for the patient, such as the development of the cancer. Both the quality of the Cas9 nucleus enzyme and the duration Cas9 expression play crucial role in minimizing off-target activity. As conclusion, transforming CRISPR system into the gene editing tool has brought revolutionary change to life science. Next generation gene editing technologies have expanded versatility of CRISPR system, providing powerful new tools to study biological systems, human disease, and agriculture.